Hey folks, I welcome you to MRM's 001. To this episode's question is, what makes dicers efficient? But before that, what is a dicer? A dicer is a ribonuclease protein, a cytoplasmic ribonuclease, and it is well researched for processing the precursor microRNA before it passes it to the Argonaute proteins. Argonaute proteins, here its short form, I mentioned in agoproteins. The process metabolism of this microRNA starts from nucleus with the drosha, another ribonuclease protein, in nuclear ribonuclease protein, and this processing completes in cytoplasm, beginning with the dicer and some other complexes, and then finally ends up with making interaction with the agoproteins. Since microRNA guided ecoproteins find their targets via base pairing between an mRNA and small region of sequence at the microRNA 5' end, that is the seed region, the main region, it's about 7 nucleotide region from guanine 7 to guanine 8, G2 to G8. So even a single nucleotide shift within that region is very dangerous. It can shift the entire repertoire of these microRNA targets. So therefore, this processing of precursor microRNA from the nucleus with the help of dicer, it's very essential for getting an efficient or getting a mature microRNA so that Argonaute can do its job. So such efficiency can be attained from dicer, not only the help of dicer, but with the help of its partner proteins. Even it is known that this dicer and its partner protein complex also ends up while interacting with these agoproteins too. So here in the screen, this is a basic architecture of the gene of dicer. Here dicer 1 is the gene of Drosophila melanogaster. See, this dicer gene basically has three domains: head, core, and base. So from starting with head domain is this ribonuclease domain, orange and yellow and green part, and the core domain is the PAS domain mentioned here. DUF, here its full form is domain of unknown function. It also has a property of binding towards the RNA. This protein has its function, and the base region is this helicase domain. This is the 3D structure of this only the dicer complex, where I am showing with arrow. This is the PAS domain, and here lies the ribonuclease domain, and at the base, that is the helicase domain. You might be wondering why I've told the head domain as this ribonuclease domain, but it lies at the center. This protein complex is arranged in such a way that it fell in a way that it appears like that only. Okay, so now beginning our next step, that why this efficiency of our dicer is being made very much efficient with the partner proteins. We will get towards the recognition, how the recognition is done, and some following steps, so that we can get the idea why this partner protein is making a dicer efficient, because itself dicer has its ribonuclease domain, which will cleave the precursor microRNA, that is converting into precursor microRNA to a so-called mature form, that is removing its loop structure. But alone, this dicer complex, although it has the ribonuclease domain, it cannot do that thing. It requires its partner proteins, so that those proteins can hold this microRNA complex into its position, and the dicer can do its job. So we'll get to understand this in a very short and pictorial way. So Before start the recognition, let us know this double standard RNA binding domain containing partner proteins, namely here in Drosophila melanogaster is loquacious, and in mammals it is known TRBP or PACT, PACT. So initially, uh, the complex dicer 1 and LOXPB basically they remain in closed conformation, uh, but tends to open while it gets interacted with its authentic precursor microRNA. Now, for the recognition part, uh, this is I mentioned you before the name of this partner proteins, loquacious and TRV or pectin mammals. So in my in this left side here is the loquacious protein, how it looks like while it tries to hold the microRNA. This is the the precursor microRNA, and the associated RNA binding domains is mentioned here clearly. And here below, this is the LOXPB. LOXPB is a loquacious gene, basic architecture of it from same the organism, that is Drosophila. So how does the LOXPB binding occurs? It's very simple here. So the LOXPB binding, it's how the order works. It's still under research. In that is the current 2022 research. It's going on. I will mention the references, the paper where I took the information in the description below. Please, you can follow there. You can read the whole article. It's very interesting. So I will not tell you that it is the mentioned order is sequential or not. Because I don't know. Even the researchers who did the work, they even don't know, because they told that they need further research regarding that. So, but regardless of that, I will discuss why how the binding happens and what they have found. So in LOXPB binding, the double stranded binding domain 3 is a third binding binds to the helices domain of dicer 1. This helps to fasten the LOXPB belt around the substrate RNA. Substrate RNA here is the precursor microRNA. Now here the double-stranded binding domain of one of LOXPB, it binds towards the PAS domain of dicer 1, and also it interacts with the RNase3 domain. Here the thing is that with RNase3 domain, it doesn't bind with it. It gets in a conformation which is like a tip to tip conformation. Okay, it doesn't bind, but it interacts in a way that it looks like in a tip to tip way. The authors of this paper, they said that this arrangement basically helps to hold the microRNA in the position where this microRNA will not move. It will keep on its way, which is necessary basically for doing the succeeding catalysis. Now, finally, the second domain of this LOXPB RNA binding domain binds towards the RNA binding domain of the dicer complex. So it is required. Sorry, so it is done in the solvent exposed site, not on the internal side, but in the solvent exposed site. Now we got to know about this loquacious binding. Now we'll get to know about dicer binding, how dicer holds this microRNA. The dicer 1 here recognizes the two nucleotide 3' overhang region of the precursor microRNA. Specifically, it's from C59th position to U60th position. These are the two initial overhang nucleotides. So this nucleotides using a pocket formed primarily by aromatic residues from the PAS domain. Yeah, the figure represents here the represents here that here this area forms a pocket-like domain. Okay, the pocket-like domain, so that this dicer uh, one space domain can recognize the 3' overhang region. The next recognition is also by dicer 1 by its uh, by its other pocket domain. The precursor microRNA 5' phosphate region is held in a pocket formed by the side chains of arginine residues and by a backbone of amide groups of alanine and arginine residues as well, which belongs to the pocket domain that is present in the dicer complex. Because I previously showed you, the core region has this PAS domain, and to the right of it is the pocket domain, and towards the left of it is the DAF domain, that is domain of unknown function. Now after that, the next thing, recognition is done from locations and from dicer. Okay, since both of them done their job recognizing thing and holding it in its own place, now it's time for this substrate to get stabilized. How does it get stabilized? The stabilization is very interesting here, because it also, because this stabilization is 50-50, like 50% is done by the holding complexes, and the rest of the 50% is for the characteristics that that precursor microRNA has. So interaction of this RNA bound domain of LOXPB with the RNase domain of dicer 1 is shown here. Here it is done in a tip to tip fashion that I mentioned previously. Secondly, the LOXPB double stranded RNA binding domain 2 and the dicer 1 double stranded binding domain stabilize the lower part of the precursor microRNA stem. This lower part is this part. Okay, part, and now the end remains with the loop of this precursor microRNA. What happens with it? Even that loop has its own essential function. The nucleotides present in the loop forms a canonical Watson-Crick base pairing, and this base pairing helps it to position itself into a pocket formed by the dicer 1 and this LOXPB. Okay, this in their interaction form makes a pocket like architecture here within this complex that can only be fit by a loop where a specific nucleotide presentation could make a canonical Watson-Crick base pairing within them. So in summary, I want to mean that the sequence that has within the loop plays a very big role in how the loop is actually formed, and this how this loop is formed actually has a role to play to get a proper fit within the binding pocket that is formed with the help of LOXPB dicer 1 complex. Now together, whatever interactions we get to know here, together all these contact positions of this LOXPB and dicer 1 complex with the precursor microRNA stem and its loop within the helps to get this precursor microRNA into the dicing center and stabilize the displacement of sizable phosphates at the 3' end and the 5' end of the prospective microRNA. So all those interactions held very helpful, and these interactions of this partner protein helped the dicer to get more efficient, although it has its own role to dice this precursor microRNA, but alone it cannot do. It requires its partner LOXPB. In case of Drosophila, So in summary, I have an illustration that I prepared, uh, making some sort of graphical changes, but mostly the figures are also from the research article only. So primarily, this whole complex remains in a closed form. This closed form here, the first, second, third represents the LOXPB domain. LOXPB domains. The first is the RNA binding, first RNA binding domain, second RNA binding domain, and this third RNA binding domain. This represents how might be sequentially this could have happened. Okay, this is a postulate situation here, because the authors say like that, and they also even discussed this limitation even in the end of their research article. So let us do the summary here. In this closed condition, the whole complex looks like that. After it gets its precursor microRNA from the nucleus, here it is represented in colored form. It still remains in closed form, but here what happens, the whole complex try to understand the overall characteristics and features of this precursor microRNA, so that it can go into this open conformation. I mean, here open conformation means into catalytically active state. But before that, this microRNA is being judged of all its characteristics by both of these proteins. I mean, the whole complex. So after that, when perfect precursor microRNA has been attained, the complex gets activated to do the catalysis. Here in this form, this loop is the whole complex try to adjust the loop into the loop pocket that the whole complex forms. So after getting in that conformation, what happens, two types of events can happen, either starting with the 5' cleavage, 5' strand cleavage, or with the 3' strand cleavage. There is the part where authors get confused, that is it, they don't know, it can happen in 5' strand, it can happen in 3' strand. This sequential order is still under confusion. So after that, it's get cleaved both in both strands. It get cleaved. The appearance of the structure looks like this. After that, the uh, loop gets removed, and the matured form of or cleaved form of microRNA gets released and looks like that. Thank you. But wait, before that, I also can give some another point towards partner protein efficiency. I got from the paper at the final moment. I got a line here, it says that these partner proteins also help to correct the suboptimal geometry of these inauthenticate substrates to make it into catalytic center of dicer proteins. Interesting, don't you see? So if an lowly featured microRNA comes into contact with the dicer LOXPB complex, so the whole complex try to adjust it into its pocket to get catalytically cleaved.